The overall goal of the following experiment is to measure chaperone-dependent intracellular protein refolding after heat shock using a luciferase reporter. First, transfect cells with plasmids coding the luciferase reporter gene and the chaperone of interest. Then split the transfected cells for recovery time points in the experimental design. On the next day, heat shock the cells to induce protein unfolding, allow recovery to regain intracellular protein folding, and then measure luciferase reporter activity. Results measure time-dependent and chaperone-dependent protein refolding based on the luciferase assay.
The main advantage of this technique over existing methods like in vitro refolding assay is the possibility of having a mere physiological readout of the effect of compounds or proteins on the chaperone activity.
The implications of this technique extend toward therapy of a wide range of diseases like neurodegenerative diseases or cancer, because chaperones play an important role in the development of these pathologies.
First, pre-warm the culture medium, PBS, and trypsin to 37 degrees Celsius in a water bath. Wash a confluent culture of HEK293 cells with 5 milliliters of PBS. Then apply 1 milliliter of trypsin containing 0.025% EDTA. Gently rotate the plate for even distribution of the trypsin and incubate at 37 degrees Celsius. Periodically, shake the plate to verify cell detachment. Now resuspend the cells in 10 milliliters of complete DMEM medium and transfer to a 50 milliliter falcon tube. Then enumerate cells using a Beckman counter.